So hello everyone, my name is Carlos. The title of my poster is Optical Coherence Tomography for Medical Imaging. I am a researcher working in Tyndall National Institute, which is part of University College Cork. So here's my poster, and we'll talk about an introduction, the methods, the results, and finally a conclusion. So think about the following situation. You have a visual impairment, you go to an ophthalmologist, and he or she tells you that you may have a problem in your retina. How would you be able to see this? Well, how would you be able to see this if your retina is approximately 25 millimeters in depth beyond your cornea? For this purpose, there is a technology called Optical Coherence Tomography, which is a non-invasive, high-resolution 1 to 15 micrometer medical optical imaging technology capable of providing cross-sectional images with a depth of 1 up to 1.5 millimeter. So in your image here, you have a commercial OCT where the patient has his eye scanned, and you can see we can make an image of the retina, we can see the internal layering, we can see its structure, and with this, the ophthalmologist has an insight of what's happening in the retina. A second application, which we're working in this project, is to combine the OCT with an endoscope probe to image internal organs, for example, of colon. So how does OCT work? Think about an ultrasound, but instead of using sound, we are using a light source like a laser or an LED. So the very basic explanation is that OCT measures the time that it takes for light to come back from a reference mirror and compared to the time it takes to come back from a sample. This is measured with something called an optical setup called an interferometer. So here's our interferometer. We have a light source, which can be a laser with this profile. The laser is going in this direction, and the beam splitter, what it does, it creates two paths, so two beams. One of them goes to the reference in this direction, then it comes back. The other one goes to the sample and then comes back. If the time that it takes is within a parameter of the laser, the photodetector will give us an interferogram signal, which is, imagine of a radio frequency that is way faster. Now, a photodetector converts light into electrons, and that's how we detect our image. So what about our results? So in this project, we have imaged rat colon tissue that is used to develop a cancer model in order to study its development. Now, OCT allows us to observe differences between what is called a neoplastic polyp or affected tissue compared to healthy tissue. So here's our sample. One region has a healthy area and the other one has a developed polyp. If we see our first polyp, we can see in point number 1, we can see like an asymmetrical growth on the surface. We can see also internal spots with different backscattering properties. And what's very powerful about OCT is that the penetration depth in this direction is about 1 millimeter with a resolution of 10 micrometer. And finally, if we compare our polyp with a healthy tissue, we can see it has a layer with bumps in the epithelium, and most importantly, we can see this internal layering inside the healthy tissue. So those parameters will allow us to differentiate between one another. So the purpose of this is to combine everything in an endoscope probe to give a more accurate information to endoscopists. So finally, my conclusion is that OCT is a very useful technology to see subsurface image data of biological tissue. And finally, OCT has the potential of becoming an optical biopsy to aid clinicians to make more accurate disease diagnosis. Finally, just acknowledge the funding, the Piccolo project, and also SFI, and Tyndall National Institute, and UCC and University College Cork. If you have more questions, you can find my email on the attached post.